Welcome, everyone. We're going to go ahead and get started. Thanks for being here for our very first research seminar of 2025. We were just talking about how weird that sounds, but we'll get used to it here soon. My name is Cinnamon Moffat. I'm the associate director of research support here at Hatfield. This is a hybrid event, so we have folks online and folks in the room. If you haven't been here before, there's a couple of tricks that we use to make sure that folks online can interact with us. So when we get to the question and answer section, if you just raise your hand, I'll bring you this mic, and that way folks online can hear us and interact with us in that way. For folks that are online, if you have any questions for our speaker today, please put them into the chat box. Our volunteer, Roseanne, hi Roseanne, will read those out, and so we can make sure we get your questions answered as well. If you are having any technical issues with Zoom, we've been doing this for a while, but every once in a while we think we know and it doesn't work the way we think. Go ahead and put those technical questions into the chat box as well. Roseanne is a whiz and she'll help you figure out whatever's going on there. But again, everybody, thank you so much for being here. I wanted to just make a quick announcement and then I'll introduce today's speaker. So our seminar series begins, like I said, today and happens every single Thursday, same place, same time. So next week on January 16th, we have Alex McInturf, who will be talking about where are the basking sharks. It's going to be a really exciting seminar, so I hope you come back for that one. If you do not get information about our seminar series and you're curious about joining us, you can go to the HMSC website, go to the events page, and there is a calendar there that has all of the information about all of our different events, and you can join us at any of them. So thank you so much for coming. But I'm just going to quickly get us going with today's seminar because I'm really excited about today's topic. So today's speaker is Dr. Richard Brodeur. He is a retired fisheries oceanographer who most recently worked with the Fish Ecology Division of the Northwest Fisheries Science Center, which is a NOAA-based group based here in Newport. He worked on habitat preferences and ecological trophic ecology and recruitment processes of marine fishes. Rick is presently affiliated with Oregon State University as an adjunct professor. Rick received his bachelor's in fisheries science from the University of Massachusetts, his master's in oceanography right here from Oregon State University, and his PhD in fisheries at the University of Washington. He has focused much of his research on feeding and food web interactions centering around fish, but he's also had a long-standing interest in jellyfish and particularly how they interact both with exploited and non-exploited fisheries. And so for today, I am so excited to learn more about some of the stuff that he has been working on. So I'm going to hand this off to you.
Okay, can everybody hear me? Okay, thanks, Cinnamon. I really appreciate everybody coming on this, one of our typical sunny winter days here. But hopefully it'll be worth your time here as I'll talk about some of the work that I've been doing sort of in the last maybe the last 5 or 6 years mostly. And I'd like to start off by saying, even though my name is the only one on this title here, there were actually a number of people that have helped along the way, a lot of collaborators from different institutions and agencies along the West Coast. So I would thank them for all the help and encouragement and support they showed. All right. So getting into the topic of the thing, I think probably all of you are familiar with what a heat wave is, at least the ones on land, terrestrial heat wave. But probably a number of you also realize that there are marine heat waves. And there's actually a definition for that. It's a prolonged anomalously warm water event that can be described by how long it is, how strong it is, how fast it evolves, and also how broadly it extends. And this is just showing an example here of, oops, let me put the pointer on, sorry. Okay, so this black line down here is what maybe a typical year might look like, warmer in the summer, cool in the winter. But occasionally there'll be years where you get temperature like this, where it's way above normal. And there's actually, because of the amount of marine heat waves that have happened recently, they've actually come up with a a sort of a classification system similar to what you have for earthquakes and hurricanes, so that there could be different levels depending on how many standard deviations the temperature is above normal. So, and these have happened all over the world. This is just an example of some of them for over a 10-year period or so. But even since this time, there have been a lot more heat waves. And you can see they show up in Australia, particularly had a lot of them in the last few years, and bad ones in Europe. But the one I'll be focusing on is this very large one in the North Pacific here, which occurred mainly in 2015 and 2016. And these are temperature anomalies, so degrees centigrade above the long-term average. So this is really warm water. And what happened is in sort of the winter of 2013 going into 2014, there was a real anomalous high pressure system that formed over like the Gulf of Alaska. And if you're familiar with it, usually that's the time where there's a real low pressure system. That's what causes all the storms and the mixing and everything. But this was really unusual to get a high pressure system set up over there. And what happened was, because there was not a lot of wind mixing with deep water and the temperature of water stayed right at the surface, it warmed up pretty quickly. And there was actually a sea surface, sort of a warm temperature anomaly that occurred in the North Pacific, several degrees above normal. And this, these authors actually came up with a very original name called the blob. So that's what it's been known as ever since. And so this blob is a very big feature in the North Pacific. And this is showing temperature anomalies from coming up here from July 14. So these are quarterly going up to August of 2017 here. And you can see the temperature, the reds and yellows are above average. So these are warm anomalies. And you can see during that period, there's a lot of red and yellow in the North Pacific, very little blue. And what this is caused by was mainly this warm blob, which is up here in the Gulf of Alaska. It started up there. And at this time, there was a little bit of cold water. You can barely see it along the coast here in the California current, but pretty much everything else is warm offshore of that. And then you can see that it spread around a bit. And then starting in 2015, there was really a big El Niño that formed in the tropics. And these move up the coast, and then so more warm water came in. So it was pretty much warm for all three of these years here. And this is for right off the coast here. There's a buoy that's about 22 miles off the Yaquina Head here. And this is the temperature. There's an instrument on it that measures sea surface temperature. And this is the temperature anomalies for 2014 from January to September here. And you can see that some days it's a little colder than others. These are daily records. And other days it's a little warmer, but not much different really. But then all of a sudden, this is when the blob came ashore. And in fact, on September 14th, 2014, it arrived at that buoy at 10 p.m. to be exact. And the temperature shot up about 6 or 7 degrees, which is really unusual. That never happens out here. It maybe changes 2 or 3 degrees sometimes, but never 6 or 7. And you can see that it stayed that way for quite some time. Okay, so this is another plot that shows temperature within this little box off the coast. So we're right about here. So if you look at this little box off the coast of Oregon and Northern California, in the black line or the blue line here is the normal temperature that you see, the long-term average. And you can see that through 2014, it was, the black line was the actual temperatures. You could see it went a little bit above, a little bit below, but it was pretty much right around average. But then during that fall of 2014, all of a sudden it really went up fast. And these are temperatures that are much warmer than normal, way out of the normal range. So you can see that it lasted quite a long time, maybe dipped down occasionally, but was pretty much above normal all the way to almost 2017. So this was what was classified overall as a severe marine heat wave, not the highest level, but pretty next to the highest. And out in the North Pacific, it lasted for 711 days, which, as you can imagine, is way longer than any heat wave you'll see on land. So that's one thing about ocean heat waves. They tend to last a lot longer because temperature, once it gets in the ocean, it takes a lot more time to dissipate than on land or in the air. And then you can look at sort of the spatial extent of these heat waves. And this is a plot showing sort of the percent of our exclusive economic zone, which is everywhere from basically the shoreline out to about 200 meters or 200 miles offshore. and throughout the whole West Coast. And this shows how much of that is covered by, so the reds are a big part of that area is covered. And you can see through here that there have been a number of warm periods. The 97, 98 El Nino was probably the strongest El Nino ever recorded up to that time. And you can see it got pretty warm over a very large area. And then continuing on, we had another warm period, early 2004 or 5. But here's what the area during the blob, you can see how much warmer that is over a really extended period area. And then you can see here that we've actually had other heat waves since then, 2019 and 2023. So this seems to be a recurring thing going on out here. So there were, as you can imagine, with all this warm water, there were a lot of ecosystem changes that happened out there. And I can give you a lot of examples, but one thing you're probably familiar with, there was a lot less productivity, but there were lots of harmful algal blooms. So they ended up by having closures of shell fisheries up and down the whole West Coast, which was a real problem. They also had many tropical and offshore species that came close to shore. So we were getting things like sea snakes and stuff that were tropical things that were found up off our area here. So really unusual occurrences. There was also a lot of changes in fish reproduction. They're timing fish that usually spawn in the wintertime, or I'm sorry, in the summertime, were spawning in the wintertime. Growth was really reduced for some species, but then increased for others. So there's a lot of change in the fish. There were a lot of what they call unusual mortality events for both marine mammals and seabirds, and a lot of changes in their distribution. And I don't know if you caught it, but there was a really good talk about maybe a couple months ago by Julia Parrish on, there was something like tens of millions of birds that died in the North Pacific because of this blob event. But the thing that we'll be talking about, I'm going to talk about today is there have been a lot of changes that were noted in the regular gelatinous zooplankton or jellyfish that we get off here, with decreases in what we normally get, but increases in basically an unprecedented bloom of these things called pyrosomes in the North Pacific. And you may be wondering, well, what is a pyrosome? Yeah, you probably haven't heard much of that. This might be your only exposure you've had to pyrosomes, that they wash up a lot in the beaches over the last decade or so. And for me, that was probably, if you asked me about 10 years ago what a pyrosome was, I probably couldn't tell you much because I had never seen one. I've never caught one, even though I've been working on jellies off of here for probably 20, 25 years. So I had no idea. I mean, I knew what they were, but I had no idea much about them. So, but I'll tell you a little bit about them. So these are your normal jellyfish that you're probably very familiar with, the large medusae that you see all the time. And then these other ones, ctenophores, you often see these on the beach. They're comb jellies, they're called. And these are sort of what you would consider your typical jellyfish that are found in coastal waters. But there's another whole group of, in this group called the tunicates, that tend to be more oceanic, more low productivity waters. And they include a bunch of different groups here. Very beautiful animals that you can see that are they're transparent like all jellies, but they have a lot of different body shapes. And if you look evolutionarily, this is sort of the evolutionary tree of all the animals out there. You can see that the regular jellies and the comb jellies are very, they come off the early branch. So they're very ancient lineage, very primitive compared to most of the other invertebrates out there. But then when you look at the pyrosomes, they're actually way up here, and the closest relative to them are actually the vertebrates. So we're probably more closely related to pyrosomes than pyrosomes are to other jellyfish. So it's kind of interesting. They actually have a notochord, which is very primitive backbone, but so they are very closely related to us and diverged relatively recently. So when you talk about pyrosomes, there's a number of species found around the world, but pretty much the most they're talking about is this one species, Pyrosoma atlanticum. And this is by far the dominant, found in pretty much all oceans of the world, mostly tropical and subtropical waters. And these have been known for a long time. The thing about these is that they bioluminesce. So this old sailors back in the late 1700s and early 1800s were describing whole areas of the ocean that would light up because of these guys. And in fact, their scientific name, Pyrosoma, and it's a Latin name, stands for fire body. So they were well known as being bioluminescent, probably one of the more bioluminescent animals in the ocean. So this is kind of the distribution as we used to know from, these are locations where pyrosomes have been caught around the world. And you can see that they're pretty much between 50 degrees north and 50 degrees south through that whole band. But one thing that's interesting, they do occur up to 50 degrees north in the Atlantic. But when you look at the Pacific, they're a lot farther south, probably closer to 30. Southern California is the most northerly they had ever been reported. Certainly well south of where we are, around 45 degrees up here. So, but that all changed, and partly because of both the blob, but also the El Nino that followed. There's a large number of these pyrosomes came into the Northern California current and created what we were calling the Great Pyrosome Plume in the North Pacific Ocean. And I'll talk a little bit about this. This got a lot of press coverage, actually. We were getting calls from reporters pretty much every day asking about, so they got, a lot of even international coverage, but their common name is sea pickles. So that's why you can see it here, but people were asking all about it. And, as it says here, the fishermen are scrambling to save their nets and scientists hurrying to study the rare animal. And I was one of those scientists because I knew very little about these guys. So I was racing to find out what I can find out about these guys. But to give kind of a timeline of when they showed up, I'm going to show some examples of sampling along the coast from, so this area down here has had a regular plankton sampling. So these are with bongo nets towed throughout the water column. And then actually their sampling started in around 2006. And what this shows is both for the onshore, so that's on the shelf, so less than 200 meters depth, and then farther offshore here, above here. And what this is showing, the dots are where they were collected during this time, and the little triangles down below are where there were samples. So you can see there were a lot of times, and if you went back, there would be zeros all the way back. So they were never collected. The first time they were collected was in the fall of 2016 on both inshore and offshore. But you can see they've been here pretty consistently, even up to this year. And in fact, there's probably a couple more dots I've heard that could be added to this figure all the way up to November. So they still are showing up here. Now, what I'd like to do is show here two different other time series that are here. There's what's called the Newport line, which is just right off Yaquina Head here. This was sampled wells continuously since 1996 or so, but sampled way in the past since the early 60s. And you can and that there were never any pyrosomes, to my knowledge, that have ever occurred in any of those samples prior to when they showed up here in blue, probably around late 2016 again. And then the other time series is up off of Canada, which is not so much a line, but sort of an area. They do have a line there, but it's sort of large within this box. And they've been sampling back to the 80s or so. And again, no pyrosomes until about the same time, actually showed up a little earlier up there than they did in the other places. But you can see that in these northern latitudes, they've died out. There's occasionally a couple of them in Newport, but not much compared to down off of California. The other sampling I'm going to talk about is a series of samples that I was involved in when I worked here for NOAA, where we go out and use like the large ships like the Shibata here to go out and sample using large nets, but very fine mesh to collect. And what we're going after are small fish, juvenile fish, and other things like squid and stuff like that. So we sampled for a number of different years with these very large trawls. And these are the areas that we sampled up and down the coast from basically the Oregon-California border up a little bit into Washington State. And, oops, sorry. So what I'm showing here are the catches of pyrosomes in these trawls. And these are the first 2 years we had in 2011 and 2013. And this is a log scale, but you can see that blue is basically 0. And this little number up here is the mean catch per trawl. So we basically got no pyrosomes those 2 years. But then 2014, they started showing up in the southern part of Oregon. And that was actually the first time I'd ever seen a pyrosome. I didn't even know what it was when I first pulled it in. And then we got them, they were found offshore in 2015. And then they really came in big numbers, 2016, 17, and 18. And you can see the mean catch for like a 15-minute tow was about 12,000 pyrosomes. So, And in fact, there were so many this year, the toes look like this. We actually had to stop our sampling because our nets were breaking from all the pyrosomes coming in. So it's just a real incredible change in the ecosystem out there. And then one of the things we want to look at is, well, what kind of conditions seem to favor pyrosomes? And what this shows is sort of the probability of occurrence. So where they're more likely is the redder ones, and a relation to temperature in this axis, salinity here, oxygen, and then chlorophyll going in this direction. And what you can see is that they tend to be found at very high temperatures. We got very few at the cold temperatures, sort of intermediate salinities, very high oxygen levels, for the most part, some exceptions here, and extremely low chlorophyll. They don't get very many of them up in the high chlorophyll waters. Okay, so then the sampling that I just showed you was sort of off the this area right up here, but it turns out that there is a group from California, from the NOAA lab in Santa Cruz, that sampled down the same kind of sampling all the way down to the US-Mexican border. So we have a pretty good idea what's going on the whole coast. And this is sort of from their sampling. And I want to point out, say, they started this one back in the 80s, and they occasionally would get one or two. This is a log scale, so there may be one or two individuals that they collected during those cruises. And you can see that goes, most years had none. But then more recently, you can see how it's changed for both their cruises and then all these other areas going up and down the coast. And in fact, the interesting thing is our area, the blues and the reds, and we actually had the highest catches of all those, going from 0 to really incredibly high catches. And again, just to show geographically the distribution. And what these show from all the way from Mexico to basically, Willapa and Washington, the catches in different colors. The reds are more higher catches. And in the mean latitude is this line, and then the highest catch is where the little star is. And you can see in 2013, 2014, they were pretty far south. But then they started moving up. By 2016, the highest catches are in our area. And then 17 and 18, they were pretty much mostly up here, very few off of California. But then 2019, they disappeared and went back down to California. So real change from one year to the next. And then we actually looked at sort of a broader scale. We looked at a number of surveys all throughout the whole Northeast Pacific. And we found them all the way up as far north as the sampling surveys went, which is up off of southeast Alaska, all the way up by Prince William Sound. It was actually pyrosomes collected way up there. So this is an incredible range extension for this species that they're usually found mainly in the tropics. So now I'll talk a little bit more about their biology. As I mentioned, they're actually colonial animals, so I'll talk a little bit about that. And the big thing about these is they feed on very small particles, very submicroscopic actually. And the thing is that their ratio of the size of their body size to the food items they eat is about 50 million to 1, which is incredible ratio. There's, like a typical copepod, I think, is something like 500 to a 5,000 ratio. So these guys are eating incredibly small items compared to the size of their body. So how they do this is, as I mentioned, they're colonial. So they're actually all these little, where these little dots are, these are all individuals. This is a cutaway through the side of one of these pyrosomes here. And you can see all little individuals. These are the light organs up above here that light up, and you can see them here in the individuals. And what they do is they bring water through here, and then they catch it on this little basket, little tiny food particles, and then they push the water out this side, and then it goes into the central chamber, and then it comes out here. So that's actually not only how they feed, but it's how they move through the water. They push themselves with all this water they keep bringing in. So kind of an interesting way to operate. And again, here's another picture. So the phytoplankton comes in, and really relatively clear water ends up by coming out. And again, the animal moves in that direction, stuff like that. But as they can clear a huge amount of water relative to a lot of other marine animals. And they also, as I mentioned, feed on very small organisms that you cannot see with the naked eye. They're so small. And I'll talk a little bit more about those. So one of the things we were interested in, since we don't know much about these guys, we actually got some money from the National Science Foundation. Actually, a colleague of mine, Kim Bernard, on campus, led the project, but a number of us were involved, where we wanted to learn a little bit more about them. So we sent out a proposal called Invasion of the Sea Pickle, and it got funded. So we had some project goals that we wanted to look at sort of smaller scale distribution, both horizontal and vertical of these guys. But more importantly, we want to look at their diet composition using a bunch of different techniques, and I'll talk a little bit more about those in detail. So we had a number of cruises that were sort of piggybacked on other cruises, mostly on large ships like the Shimada, during February, May, and August of both 2017 and 2018, which you recall, those are really the 2 big years of pyrosomes. And then, so this is some of the results of some initial results that we got. So this is showing the length distribution. The Februaries here are in dark color, and then the greens are in May. And you can see that there was basically a doubling in average length. So they went from something about, well, during the wintertime, this is about the smallest size that we see them, to something like sometimes by the end of the summer to be about 1 foot long. So they grow pretty quickly. But the thing that was interesting is that the densities of those abundances didn't change at all between these 2 time periods. And this is kind of just showing the general distribution of where we caught them in the May cruise, and then the August cruise was mostly up here off of northern Washington and up off of BC. And it's showing again, sort of plots that show the environmental conditions we saw them in. So temperature, they're definitely found at higher temperatures. They were not found at all low temperatures, kind of mixed with salinity, but highest were sort of intermediate. And then the big thing was chlorophyll, real drop off, although there wasn't significant in terms of the relationship. There was really sort of a knife edge thing around this 2 fluorescents here, but they just were not found above that. So they were only found in much more clearer water down here. And if you look at their vertical distribution, we actually put some GoPro cameras on these CTT things that went down and collected water samples up and down the coast. So here's an example of what they look like when you looked in the cameras. You can see all these little things are pyrosomes. And we found that their highest densities, at least during the daytime, was around between 49 and 68 meters. So well below the temperature where it changes, what they call the thermocline, where it changes the most, but above where the salinity changes the most, and definitely not in where the chlorophyll is the highest. So they don't like waters, it seems, that are very high chlorophyll. So we wanted to look at their diets next. And as I mentioned, you can't just take out, well, they don't really have tummies, but you can't really dissect them and take out their stomach contents because their things are so small, you wouldn't see it with a regular microscope. So we've used some other techniques. And this project was done in collaboration with Ann Thompson at Portland State up in Portland, where we looked at using flow cytometry, which is a way to look at sort of the size of the particles. And these are 1 micron beams. So along this axis is the size of the particles. And the things that they were eating were all well below 1 micron. So these are tiny things. And then we also did some environmental scanning electron microscopy and saw that there were very small parts of phytoplankton and also things like bacteria that they're eating. So really, really fine particles. So very unusual. And then another way we looked at it is by looking at biochemical markers. So we use these things called fatty acids that you can take like a little bit of tissue of the body, and it can tell you based on these fatty acids what that animal ate in the past. And when you do that, we found that for the 2 cruises, May and August, we found that they were feeding mainly on dinoflagellates, a fairly small phytoplankton, but mobile phytoplankton, and then bacteria, and a little bit of these, which are coccolithophores. So they're really, again, mostly very small organisms, as we would expect. The other thing we wanted to do is use stable isotopes, and this doesn't tell you exactly what they're eating, but it tells you sort of their relative trophic level, and then where does their food come from. And there's 2 different things that we use: carbon, carbon, and then nitrogen. And if you measure in the body tissue of these guys, you can see that where you get carbon, you'll get sometimes a different signature if they were feeding in the coastal waters versus offshore, or even like in a river, you get a really different signal. So this will tell you where they came from, or at least where they were feeding before they got caught. But also looking at nitrogen, that gives you an idea of the trophic level, because it goes up a certain level every time, or a certain amount with every trophic level. So we can use this tool to kind of look at where they were feeding, but also what trophic level they're at. And this is showing for, this is color coded by geographic area. So north of 44, which is around here, a little bit between 44 and 43, which is kind of right off of Walport here, and then south 43 down to the Oregon border. You can see that there are some differences. Basically up north, they're feeding much more on onshore production rather than offshore, what the other two had. But the more interesting thing is to look at their the trophic level, which is along this axis here. And you can see that here is what a typical copepod is. So these are like the main grazers in the ocean. And you can see that these pyrosomes, for the most part, are feeding at a lower trophic level than even copepods. So that's why, because they're feeding on very small particles. And this is where a typical phytoplankton is. So they're feeding, some of them are feeding at almost the same level as what a phytoplankton is. So that's really amazing to see a large animal like that feeding at such a low trophic level. And then the last thing we wanted to look at was sort of their ecological impacts. What, having all these pyrosomes out there, what are they doing to the trophic levels below them? So one of the things we did was do some ingestion rates, how much they feed on, looking at their gut pigment, sort of as a proxy for how much food they're taking in during one survey. So what we did was we measured them throughout the different times of the day. They tended to feed a little bit more during the daytime than nighttime. And then we looked at we kept some alive on tanks on board the vessel to look at how long it takes for the food to go out of their guts out to back out again, and it was pretty quick. And then so we use these to calculate sort of consumption at different latitudes. So these are different transects here. And then we can compare that to what's out there in terms of the environment to come up with sort of a grazing impact, how much they consume per day. So the results show that it varies by latitude here, but it goes from about 1% of what's out there per day to up over 10% some places. So that's a pretty big impact to be consumed by just one organism out there. So now the rest of the story here is going to be like, where do all these pyrosomes end up? We know there's millions of them out there, billions probably. Oops, I'm sorry. So we had some estimates of just incredible biomass of these guys off the Oregon coast and densities. I mean, we get 5 individuals for like a 1 meter square cubed on average. So that's a lot of individuals for such a big organism. We know that their caloric content is kind of average. It's higher than a lot of other invertebrates, but it's actually well below the range of a number of fish. So they're probably okay for things to eat, but they're not the best things. If they can eat fish and other things, most things would be better off. And we consider that most of this biomass either sinks down to the bottom and gets remineralized and decays down there, or it could be consumed by predators both in the water or down on the bottom. So we'll talk a little bit about some of this. There were certainly a lot of them that ended up on the bottom. This is just right off the shallow water, but also in very deep water. This is around 1,200 meters. You can see all the pyrosomes here in Quinault Canyon. A number of them ended up on the beach, as I mentioned, and it adds to that ecosystem. There's a lot of animals that live off that, animals that end up on the beach like that. We also know that there were a lot of invertebrate predators on feeding on them, especially down toward the bottom. I was very lucky to be on a cruise where they had an ROV that was diving, as mentioned, in Quinault Canyon, and we just saw all sorts of animals feeding on them, such as these sea urchins down here, sea anemones, and even a deep sea jellyfish that was hanging right above the bottom that had a pyrosome in its tentacles. So there was a lot of things that probably consumed them. Unfortunately, we don't have any quantitative estimates of this. But one thing we do have is that there are a lot of studies that have looked at diets of different vertebrate predators off the coast here. And we know that from both anecdotal evidence, but also lab stuff, that there were a lot of things that ate pyrosomes. Some examples of things that are pulled out of sablefish stomachs. Here's a rockfish that was just absolutely packed with pyrosomes. And there was also a whale that washed up, a fin whale off the coast of Washington, and its stomach was absolutely loaded with pyrosomes. So there are a lot of things that are capable of eating them. but whether they do or not is a question. So one of the things that we have, some of our colleagues at the in the FRAM group over here have been looking at diets of ground fish. So these are fish that are found near the bottom. Going back, they do a bottom trawl survey pretty much every year. They've had diets going back to 2004, I think. But I'm looking at just since 2015, since the pyrosomes came, this is all fish that had a substantial number of pyrosomes in the guts, and showing the years that they were observed and the percent of the weight of the diet during these times. So 15% for sablefish is pretty substantial amount of pyrosomes. So, and this other one had even a thorny head, which is more deep water, had even more. So there are some fish that did tend to consume them. And this is showing a plot of the catch of pyrosomes, and this is a sort of the scale here from all the trawl surveys. So these are in the bottom trawls that come up with the trawl, and that's the color in the background. And you can see that they're relatively early. There was only a few of them in 2014, mainly down south. They picked up a little bit here, but by 2017 and 2018, huge numbers right off the coast here that were found in the trawl survey. And then the little circles that are overlaid, I'm not sure if you can see them there, but the size of the circle indicates how much the diet of these fish was made up of pyrosomes. So the largest circles are a pretty high percentage of their diet, greater than 75% of the fish had pyrosomes in their diets. And you can see that corresponds really well with the with what was found in the bottom trawl survey. So another way that we saw there was a lot of interactions with fisheries. Unfortunately, we didn't get a chance to quantify that, but we got lots of complaints from fishermen about these guys fouling up their nets, like in their troll gear. And anything that towed a net was full of pyrosomes, and it really affected the catches. This is an example from shrimp trawls. You can see the little tiny red dots are the shrimp here, but all those brown things are pyrosomes. So you can imagine how much more work it would be to go through and sort out all the pyrosomes just to get to those few shrimp. So it ended up by costing the shrimp industry quite a lot of money those years when they were really abundant. Ooh, this is not going down. Okay, hold on. Okay, well, the other thing that was an issue for us, I had mentioned that we had trouble with our sampling. It did affect a lot of cruises, research cruises. I mean, we were getting a tow like this, which is basically all pyrosomes in the caught end of the net. And we'd bring up, totes and totes of these guys. And it really impacted our sampling because we could not, we had to go through all this to find the little tiny fish that we were looking for. So it really added a lot to and made it a real big inconvenience for us. So now we're going to get into sort of more the modeling part here. But this, one of the things is because we've had all these cruises and a lot of data on diets and things, we've been able to construct these food webs of the sort of the Oregon and Washington coast here. And this is an example of one that we published a few years back that was sort of a food web, sort of an average food web for 2000, I think it was 2006 to 2009. So this is what the food web looks like normally with increasing trophic level along this axis here. So you got the plankton on the bottom, phytoplankton and other things, all the way up to the highest trophic levels, which in this case are humans, or troll fishery and recreational fishery, but also you get marine mammals and other sharks up here at the top. And what this is, this particular food web is centering on large jellies. So our typical jellies that we get, the medusae out here. And it shows the amount of where all the things down below them, mainly there's all these things in green, the connections to them. So the more green it is, the more important these things are to them and the more connections. And then red is sort of what things eat jellies. So you can see that there were some things that eat regular jellies, not a lot compared to other things, but they are important in the ecosystem. And then this is what it looked like after the marine heat wave. So postdoc Dylan Gomes re-parameterized the whole model based on results from the more recent cruises and diet studies. And this is, in this case, we're centering it on pyrosomes here. And you can see a big difference that pyrosomes have relatively few things that they consume, but and very few things that are eating them. But some things are very important to them, but a real difference in terms of their food web. So one of the nice things about these models is that you can take one, like the one on the right, and subtract it from the one on the left, and you can see what the differences are between these two time periods. And this is sort of an example of that differencing and stuff. So in this case, the circles that are in pink indicate things that increased, along with the red lines. Those are flows that increased after the marine heat wave, versus blues are things that went down after the marine heat wave. And you can see that a number of things changed. I mean, the things that went down are basically things like large jellyfish, krill, euphausids. Those all decreased. There were a few things that went up, but really the big thing is are pyrosomes, which is that one right there. You could see what a difference it meant. Basically, it went from nothing to a huge number of pyrosomes. So that is really driving what the changes are in the ecosystem between these two different pre-marine heat wave, post-marine heat wave. So the other nice thing about models is you can sort of run little experiments or do scenarios with them to see what would happen if you changed the flow from one box to another, or increased one thing or another. And we did that. And one of the things, oops, sorry. So this is an example where we did some simulations where we took pyrosomes and we reduced them by, in the case of the blue areas here, 100% for what they had previously, all the way up to increasing by 100%, which is in the red here. And this is showing the effects on these different sort of higher level, trophic level anchovies, chinook, small jellyfish. You can see all of those did poorly when you added more pyrosomes, and then basically pyrosomes are either competing with them or somehow taking food away from them. So they all decrease. But you can also see that there's some things that increase that actually did better, such as dogfish, squid, and of course sablefish, which, as I mentioned, is a really big predator on it. They just took off during this pyrosome bloom, really high numbers. So that's kind of interesting that there's both winners and losers. And you can kind of look at all the major groups in the food web here. So it's not all the different boxes, but they're sort of combined together. And you can see, so what this is showing is if you removed all the pyrosomes out of the bottle, this is what happens to all the other groups. And you can see that a number of them did poorly. But there's also a larger number that probably did better not having the pyrosomes in the model. So there are both winners and losers in this. One of the things that to me was most interesting is that if you look at the ones that are the biggest winners by having the pyrosomes there, these are the mostly things that are near the bottom. So they're either feeding on the bottom or actually living on the bottom on this case here. Whereas the ones that did the worst at the other end are all things that are up in the water column. We call them pelagic, but they're swimming around like tunas and salmon and forage fish and then other jellyfish. So there seems to be a real trend here where the things down the bottom did better with pyrosomes, whereas the ones up in the columns did worse. And the other thing I want to point out is this here, which is fisheries, and this combines all the fisheries. So that includes things that are in the water and things that are in the bottom. So that's why you're getting such a big spread there, I think. But what we wanted to do was kind of modify this and just look at things that are like in the case of animals that are just found either in the water or down on the bottom. And this is showing when we remove all the pyrosomes, the ones on the bottom did a lot worse, whereas the ones up in the water did a lot better. And you can see that in that case. And then the same thing with the fisheries. So benthic fisheries like crab fisheries, when you remove the pyrosomes, they did a lot worse. Whereas the other one, in terms of the pelagic fisheries, is kind of even, but there are a lot of higher values up here. But it was kind of a mixed bag. But it was definitely worse for the benthic fisheries to have all these pyrosomes. Or I'm sorry, it's better to have the pyrosomes in there. When you remove them from the model, it did worse. So, and those were both highly significantly different from each other. Okay, so just to kind of wrap up things, we see that pyrosomes are found in very warm, sort of less productive waters, more oceanic waters, which is much more similar to their tropical habitat that they're normally found. And these waters are generally not found in our area, but because of a combination of the warm blob and the El Niño, they really did warm up to habitat that was much more similar to what they're found, and which led them to both come up into our waters, but also last through sometimes several years up here. So it was a really big change. we saw that they also consume very small plankton, but these are in turn fed on by some fish and other top predators, probably not as much as other things in the food web, but they're not basically a trophic and dead end by any means. And we see that perhaps we think that pyrosomes are actually helping out the demersal food web. So things like flatfish and other things that are associated with the bottom probably are going to do better. Certainly crabs have been doing better since this happened. So we think that there is a benefit with all this energy going down to the bottom and enriching the diets of those animals, but they negatively affect the ones up in the water column. And this is kind of just a little schematic that shows kind of a typical food web off of here, where you get your phytoplankton that leads to the small zooplankton that consume these. And then those have different routes, but they can go up to forage fish, which are often the main food of these top trophic levels. But during the pyrosome bloom, you have sort of a totally different food web where it's a much more direct food web. It goes from very small particles to pyrosomes to fish. So there's basically one less step, at least in the food web, but also it's a real difference because these fish tend to be found mostly toward the bottom, and then a lot of that ends up in the bottom as to help the benthic fauna or detritus down there. So a real change, fundamental change in the food web that we think is associated with this pyrosome bloom. And just to kind of reiterate, these are the animals I was talking about. These the animals that are related to pyrosomes that are also related to vertebrates, and they tend to be all like they all have mesh filter feeding like this, so they can all feed on fairly small particles compared to most other plankton out there. But they're very important in terms of nutrient cycling in the ocean because a lot of what they feed on can drop down as part of fecal pellets down to the bottom very quickly, or in the case of pyrosomes, they become when they die off, they all sink down the bottom, and there's like a real input of carbon down to the bottom ecosystem. So they can be very important, much more maybe in terms of the cycling than some of the crustacean plankton that we're more familiar with. So although we don't take these into account a lot, they can be a very important part of the ecosystem out there. So I guess the last question is, are these going to be permanent residents of the Northern California current? Well, I guess this is pretty much anybody's guess. Nobody knows for certain. I would say it's a likely possibility, unfortunately, maybe. And the reason I say that is because if you look at some of the modeling that's been done, the climate models, and depending on which model you use, the different emission scenarios, they're showing a tremendous amount of warming throughout the world's oceans by the turn of the next century here. And this is showing the proportion of the ocean with a permanent marine heat wave. So that's 365 days a year compared to the baseline or now temperature. So there's going to be a lot of heating up if these models are correct, no matter which model you use. And they think by around 2040, much of the North Pacific, shown here in this light green, is going to be in a state of permanent marine heat wave. So this is going to be great water for pyrosomes, plus a lot of other tropical animals, probably not so good for salmon or, other things, you name it. So I think we're in for some really big changes going in our ecosystem, but, things could change in a different way, but chances are this is where we're headed, unfortunately. So with that, I'll end with this is my favorite side of a pyrosome. This is actually a tropical pyrosome, not the one we get here. But this particular one can grow to about 30 feet long. And it's big enough that a diver can swim inside the hole and turn around and come back out. I mean, they're huge. Fortunately, we don't get these here, but they turn out to be a very favorite food of, in this case, the leatherback sea turtles. You can see them riding this like a little water wings going on but actually probably consuming this guy here. So with that, I think I will be happy to answer any questions. Thank you very much for your attention.
Thank you so very, very much. I appreciate that. There was a lot of information in there, so I bet we're going to have some questions. So I'm going to take turns between folks online and folks in the room. Roseanne, any questions online? Okay, for folks online, go ahead and put any questions into the chat and we'll get them read out to our speaker today. How about questions in the room? Oh, yeah, there they are. 1 second.
Nice talk, Rick.
Thanks.
My question is, how much control do you think they have in terms of how far they can swim? Thinking about invasion, but in terms of thermal preference over maybe changes in currents or things like that?
Yeah, that's a great question. Yeah, they can swim quite. In fact, I think in the tropics, I've read papers, they can swim like vertically up and down the water column about 700 meters. So they, in a day. So they can move pretty quickly if they have to. When it comes to sort of the larger horizontal currents, they probably could swim in a particular direction, but I think they're probably just infected in with the prevailing currents that come in. So like when the El Nino current, generally they come from south to north up our coast. So these currents often bring lots of warm water animals that can't swim that well, but as well as some that can. But I think the pyrosome probably just were transported up for the most part. I'm not sure they would know to orientate and swim north. They probably wouldn't want to, but so I think they were just infected up into this area. Good question.
Hey, Rick, that was awesome. So much fascinating information. You mentioned when they die, they're a really important benthic input. But what makes them die? It seemed like it was all at once. You had tons of pictures of hundreds of them on the seafloor.
Yeah. Yeah, well, we don't know for sure. Probably some of them die of old age. They don't naturally live to be probably more than a year. But I think what happens is we often see big mortality events after when there's a big phytoplankton bloom. And I think in that case, there's so much phytoplankton in the water, they can't filter it. It just gets clogged up in their little tiny little pores that they try to filter, their little mesh. And I think that probably they starve to death. They can't eat food after a certain point when it gets too productive. So that's why you don't tend to find them coast shores anywhere. They're usually out in blue water out in the ocean, where they do best.
I see a question online. Go ahead, Roseanne.
A great presentation. What was the final fate for all those pyrosomes that ended up on the beaches? Did they just decay, or were they consumed by amphipods and other critters?
Probably a little bit of both. I imagine. We actually tried to do some experiments on their decay times where we'd have them in the lab and see it depends on temperature and a lot of things. So they can break down in a matter of like a week or two to nothing. I mean, basically they lose that tunic, it disappears, and then all the little guys eventually dissolve, or they're, consumed by bacteria. So that is definitely one way, but I think also a lot of animals on the beach probably ate them. We really don't know. And I think that would be a good little study to figure out how much actual stuff is coming on the beach, because beaches usually do not have a lot of productivity on their own, sandy beaches certainly. So it is really a big influx of productivity onto those beaches and probably supports a lot of different animals. But I don't think we really, I don't know if anybody's ever looked at that with pyrosomes before.
Great, thank you. Questions in the room?
Hi, Rick.
Hey, Brett.
That was a great talk. I'm curious about the other appendicularians and stuff, like the doliolids and stuff. Did they increase in abundance too, or is there some reason they're not ever?
Yeah, that's a good point. Sometimes salps increase quite a bit, but not as much as pyrosomes. And salps have increased, like, remember I talked about the war period, it was a 2004, 2005. There were a lot of salps during that time. In fact, there were so many that on the California current that there was a nuclear plant down in California that they had to close it down because the intake got so clogged with these salps. I mean, there were billions and billions of them. So there have been times where salps, it's not unusual to have big salp blooms, especially after the upwelling cease and they come a little bit closer to shore. But as I said, pyrosomes is just totally different. We don't think we've seen anything like this, for sure. And, there's been sampling at least since, I don't know, here off of Oregon since at least the 60s, maybe even earlier. And they've never been reported this forth.
All right, question online. Go ahead, Roseanne.
Are all of the pyrosome life stages where the pyrosomes are? What's their life history?
Oh, okay, that's a good question. That's another thing we don't know a lot about, particularly for this species, because it's hard to keep them alive in the lab, and there are not a lot of labs, marine labs where they're found in high abundance. So, but we know that they do have, they have both what's called sexual and also asexual reproduction. Each individual is hermaphroditic, so it has male and female sex organs, so they can self-fertilize. And they start off as a tiny little, I think it's a cluster of 4 little of those, what they call zooids, those little animals together. And then they keep getting bigger, and they actually add on in a ring like this. They keep going and going and going until it'll get to be something that size. So, but I think we don't know exactly when they reproduce, probably sometime in the winter or fall, but we just don't collect here, at least the small individuals. So they're probably reproducing further out to sea, and we get them only when they come closer to shore. I don't know if that answered the question, but and as I said, I think they live probably maybe a year, maybe if they're in an ideal environment, maybe a little longer. I'm not sure of these guys though, if they can get to be that big after a year. That would be incredible growth rate. These guys probably live multiple years like that.
So I'm going to ask a question. So the growth that you were talking about, where they start out small and then they get larger, that's zooid addition and not zooid growth?
That's right, but pretty much all addition. It just kind of keep adding on, add more whorls, and it goes up until the end. And the interesting thing about them is that they're, I had mentioned about their bioluminescent, and it's interesting because they actually use the bioluminescence to communicate between the zooids, the individual little animals. So sometimes if one on the, say the closed end needs to communicate to the ones on the other end, it'll start lighting up, and then it kind of lights up as a whirl like this. And it's really cool. I've never seen them do that, but I've seen videos of them. It's pretty amazing to do that.
That's great. Okay, Roseanne, go ahead.
Let's talk, Rick. Any evidence of leatherbacks eating pyrosomes in their critical habitat off our coast? Also, a follow-up about pyrosome swimming. Is there any evidence of cues that they may use for directionality?
Oh.
Seems there may be directionality with light, possibly a Diel cycle, but anything else?
Yeah, well, leatherbacks certainly do feed on them. And I think there is I've heard anecdotal stories about them feeding, but there's not, unfortunately, a lot of studies during this time period in leatherbacks. We'll get leatherbacks that wash up on the shore in different places, but sometimes by that time they wash ashore, they hadn't been feeding for very long. So it's really, there's not a lot of data, but we know that in other parts of their distribution, leatherbacks and actually probably all the sea turtles feed on pyrosomes as well as other jellies. So yeah, they're probably very important. They're actually not in our model because turtles are not that common here compared to the other things. So we kind of left them off the model, but yeah, they're probably certainly one of the predators out there. And then as far as their orientation or what they use as cues, well, we know, as I mentioned, they vertically migrate up to 700 meters in some cases, but in our region, we did some day-night differences. It's more like maybe 30 or 40 meters that they go up and down. And we think that they come up towards the surface to feed at night and then go down deep during the day to digest or whatever. But we don't really have any behavioral data on them. It's just what we suspect based on the data.
I have a funny feeling we could ask questions forever because there's a lot of questions that are there, but we're up
Oh.
against time. I just wanted
was
to
there.
give everybody a chance to say thank you one more time.
Thank you. Thank you guys.
And then I wanted to just give you a chance to say, what's next? Yeah. So what do we think's happening next? And what other studies are going on with this continuing pattern that we're seeing?
There's probably not that many studies going on with pyrosomes anymore. Well, as you can see, they've kind of dropped off. Even though they're still out there, they're not as important as they had been in the past. So there have been studies going in other areas, but probably we're not going to be doing much here because we can't collect them as easily anymore. But there's certainly a lot of things. I think one thing I'd like to do is figure out, based on the size of them, you can use these sort of oceanographic models, circulation models, that you could sort of look at their size and kind of back calculate where they might have come from, like where they reproduce. I mean, that's one thing you could certainly do. I think the models are out there. I don't know if there's enough data at different sizes because we don't get them, much smaller than this. So, but that would be one thing. I think that's really what we don't know that much about their life cycle, where they're reproducing. That'd be an important part of it.
Nice. Thank you very much. For everybody in the room, if you didn't sign in as you came into the room today, if you could do that for me, that just helps us keep track of how many people are here. And I hope to see you again next week. And one more time, thank you so much, Rick. For folks online, thanks for being here. We'll see you next week.